more than they use to...
Possibly. Possibly. I don't know, but possibly, because I may give you a stain and then I say, what is the modern use in this staining process? I may tell you that this is a spore stain or identify what is the staining and then what is the modern which is used. But there will be choices in the question because it is all multiple choice. It's all multiple choice. One thing I will not ask you, which bacteria showed positive, say, which bacteria showed the positive amylase test? No. Okay? Oh yeah, I'm evil, but not that evil. At the same time, I expect you to know some of the bacteria, like E. coli is very specifically, it gives a green color, green metallic sheen. That's only E. coli. I expect you to know about the E. coli, but for the other one, if I show you a picture of the starch hydrolysis, and I tell you this is bacteria A, bacteria B, or bacteria C, then you have to identify which of the three bacteria is positive for starch hydrolysis based on the pictorial representation. Does that make sense?
But you haven't asked for specific bacteria?
No.
Okay.
No.
And along that same lines for like catalase tests in terms of the bubbling.
Yeah, it's the same line. It's the same line. You don't have to know which bacteria showed positive for catalase. But if I show you a picture with the catalase, bubbling, I may ask you, like, is this bacteria positive or negative for catalase? Does it make sense? If you have gone through the study guide well, you will be able to, and you will love this exam. Because it's pictorial, you say all hands on what you have done, and some people, they do really good in the lab practical exam, just because, like some people are kinesthetic learners. I would strongly advise you to go through the lab sheets, go over the lab sheets, and also do the, just follow the study guide, which I'm sure you all have done it and you all are prepped. I believe you all don't underestimate yourself, folks. You all are very good. Yes?
Speaking of the study guide, is it just 19 through 24 on this?
Oh, holy Christ, no, it's the whole 1 through 24. What, you thought only the remaining
Well,
portion?
yeah, because lab 1 you said to just go to 19.
Yeah.
So I was asking.
But the lab exam 2 is a complete one. The same study guide, it again is repeated.
Okay, just double check.
Okay. Yes.
Will we lecture on Wednesday?
Yes, there will be some lecture after the lab exam.
So it's the test first and then...
Yeah, it depends. I have not decided. It depends how am I able to cope up with my Tuesday, Thursday class. I want to keep both the lectures, Monday, Wednesday section and the Tuesday, Thursday section synchronized. So for that part.
How many questions?
Expect about 50 questions, possibly 1 or 2 questions extra credit, possibly. I'm not promising you, but possibly.
So, I'm curious. The lab exam during lecture?
Lab exam is during lecture. During the lab time, you have to still come. The 3:30 section have to still come. You have to evaluate those results, and that's it. Possibly, I will not be starting a new lab, but then you have to evaluate those results. What you studied today. Yes, Eric. No, I'm sorry. Colin, ask me those questions.
Do you mind if I ask you about the TSI?
Yes.
So I'm curious as to why the slant can be yellow when that is a fermentation of lactose and sucrose, but that's an anaerobic process, but at the slant there's oxygen present.
When there is oxygen present, yeah. Okay, now you're talking about this one, why the slant, when the oxygen is present, why the slant is yellow?
Right.
Okay, that's because some bacteria they tend to utilize the glucose by aerobic respiration. But once the glucose was exhausted, it switched to using the lactose and the sucrose. When it utilizes the lactose and sucrose, it ferments it. Okay? It ferments it, and during fermentation can happen in the presence of oxygen or in the absence of oxygen. It doesn't bother the bacteria.
But I thought fermentation was an anaerobic process. And
Fermentation is an anaerobic process, but if the oxygen is present, they are not going to utilize that oxygen. It's not going to utilize oxygen, but oxygen present is not going to bother them either. Okay? So when it grows on the slant and it gives a yellow color, it means it is undergoing fermentation. Because in fermentation only, it generates a lot of acid. And when you talk about the respiration today, when I further continue my lecture, it will be all clear. And when it generates those acid, there's a pH indicator which is incorporated into the medium. And that pH indicator turns yellow in color under acidic conditions.
And so for clarification, the glucose fermentation is happening in the butt area below the slant while sucrose and lactose is on the slant?
No. Sucrose and lactose could be in the butt area depending on the bacteria. It could be in the butt area as well as in the slant area. The butt area creates an anaerobic condition. The butt area creates an anaerobic condition. There's no oxygen present. So invariably in the butt area, because it's either the anaerobic respiration or the fermentation can only occur.
So once you start using the lactose,
So let's put it this way. If it is, so yellow in bud, it invariably means there is anaerobic, sorry. So when you have the yellow in the bud, it means fermentation of the sugars.
Of all three sugars?
It could be possibly all three, but absolutely fermentation of sugar, lactose and sucrose, and depending on the bacteria, maybe glucose. But remember, glucose is the preferred energy source, so oftentimes glucose is first used up maybe by aerobic respiration, and once the glucose is used up, then it will switch to using the lactose and the sucrose.
Is the amount of glucose almost negligible because it's so little?
It's so little compared to the lactose and sucrose because your main job is to find out whether the bacteria can utilize glucose and then switch to lactose or sucrose, or once the glucose is exhausted, the bacteria switch to using the peptones in the medium.
And to identify if the bacteria is utilizing glucose is based on the color?
It's based on the color. Everything is based because glucose is in such minute quantity. Once the glucose is exhausted, the bacteria will have either a choice to use this or to use the peptones. If it uses the peptone, then you will get this strawberry red color. Yes?
So there's a question in the lab sheet about the uniqueness of this medium. Is it that there's a very small amount of glucose and then there's the lactose sucrose, or is it that...
The uniqueness, that's one of the uniqueness of the medium, and the medium can also detect the anaerobic respiration.
Okay, because like I thought maybe it was that it tells you whether, because you have both aerobic and anaerobic abilities in it. So is that answer incomplete?
It is usually the uniqueness of the medium is because it has 3 sugars incorporated into the medium, but the glucose is in a much, 1/10 the concentration of the lactose and sucrose.
Would the glucose typically be a much larger concentration
Uh-huh,
in medium?
In the regular medium. But in the TSI, it is 1/10 the concentration, and in addition to glucose, it has also the sucrose and the lactose.
And sucrose and lactose are not usually included in mediums. in
No,
mediums.
unless until you are trying to see if there's a fermenting bacteria, otherwise there's no need. Because glucose is always a preferred energy source. Given the choice between glucose, sucrose, and lactose, 90.99% of the bacteria will utilize the glucose first. Some bacteria may ferment the glucose, but 99% of the bacteria will first utilize the glucose, and then it will switch using the sucrose and lactose if they are genetically programmed to use the sucrose and the lactose. Yes, Andrea.
So number 2, it's basically that color because of fermentation, correct?
The number 2 is this color because of fermentation of the lactose and sucrose. You are very
And
correct.
number 3 is because of the use of the peptones. But what about number 1?
The 3 one, can you tell me, is you are right. So what's happening in number 3? First, glucose is used up. It's not being able to use the lactose and the sucrose. So what are its choices? It starts using the peptones. And then, can you tell me why is the strawberry red color? Can you tell me, first of all, if I give you this, why is the red color here?
Aerobic respiration.
Is it because of? In this red part is because of aerobic respiration. Remember, but is an anaerobic condition.
Is it the deamination of the
Huh?
nitrogen? I thought it was
It's because of basic. Why?
Because when the use of peptones
The peptones. Peptone use. What does it do?
deamidize it?
Oh, there is deamination. Yeah, you're very correct. So there is deamination. And then?
And then, for some reason, it turns basic.
Oh.
I don't know the next step.
Okay, never mind. So do you all agree the ammonia which is generated here, this ammonia combines with the water to form ammonium hydroxide. It combines with the water to form the ammonium hydroxide. Now tell me, ammonium hydroxide is a gas or a liquid? Huh?
It's a gas. No, it's a liquid.
Ammonium hydroxide is a liquid. Ammonia is a gas. Ammonium hydroxide is a liquid. So once it combines with water and form ammonium hydroxide, guess what happened? The ammonium hydroxide, can it evaporate off? Can it evaporate? Nope. So what are the choices of the ammonium hydroxide? It just dribbles down. You make it? That make sense? It just dribbles down. And once it dribbles down, it will create a lot of alkaline condition, and that's why the medium turns to strawberry red. There is no growth of bacteria. There is no anaerobic or aerobic respiration in the bud. It's just because of the ammonium hydroxide formation in the slant, bacteria growing in the slant area. It simply comes down, and that causes the red color.
How do we account for the blackening in the slant, or is that just the photo?
The blackening in the slant, look at this. You see the blackening in, let me do one thing. Let me, 1 second, pull this thing out. This is getting too much blackening. Okay, now you see the blackening here is in the slant area, but here is in the butt area. Is that clear?
Yeah.
Now, if it is in the slant area, what does that indicate? Aerobic respiration using what?
So isn't there a sulfur component on the amino amino acid structure. back
And then what happens? So if you see a slight blackening like what you see over there, what it is indicative of? Respiration or anaerobic respiration? Like aerobic respiration or anaerobic respiration? But what do you, why, when you say this blackening, you say it is anaerobic respiration.
Yes.
But that doesn't hold good over here, am I correct?
Correct.
Why?
It's on the surface using oxygen.
Huh?
It's on the surface using oxygen.
It's on the surface, but then why do you see the blackening?
Because that amino acid had a sulfate. Oh wait, no, this is separate.
No, go ahead.
It had a sulfate on its body, or what's that called?
It's a sulfur-containing amino acids.
A sulfate-containing amino acid, and then that reacts with the iron in.
Why?
Can I look? It's right here.
Okay.
Because iron sulfide combined with
No.
hydrogen.
No.
Okay, I give up. Pass.
Yes, Jessica?
So sodium thiosulfate is reduced and,
In this one?
no, that one, no.
Anybody else who can throw some light? Yes. Alex? Alex? No? Anybody else? Yes. Denise?
Isn't it because of the sodium? Like it breaks it down into iron sulfide.
Oh no. Why do you see this green color, this black color?
Isn't the sulfate reduced to H2?
Yeah, and then, okay, why the sulfur is getting reduced to H2S?
Because that's
Like
part
you're-
of the respiration.
Giving a box.
Class? Then never mind, I'll tell you. What happens is, first cells uses the glucose. Once the glucose is exhausted, it starts using the peptone. You were correct up to that point. And then when they uses the peptone, in the peptone, there are some sulfur-containing amino acids. So if the peptone is, say, 10 amino acid long, up to the 5th amino acid, it is okay. The 6th amino acid is a sulfur-containing amino acid. So that sulfur atom has to be removed. That amine group has to be removed as ammonia. If there's a sulfur atom present in the carbons in the body of the amino acid, that sulfur atom has to be released as hydrogen sulfide. You see that? And whenever there is hydrogen sulfide formed, there is iron sulfate in the medium, it would immediately combine with the iron sulfate to form iron sulfide. And that's the reason why you see a slight blackening, because of the formation of iron sulfide here. But this blackening is because of the sulfur-containing amino acid which releases the sulfur. Whereas the blackening here, what you see, is because of the hydrogen sulfide that is formed due to reduction of the sodium thiosulfate in anaerobic respiration. So here there is anaerobic respiration occurring. And because there is anaerobic respiration occurring, that's the reason why it is all black in color.
So, but it has a little bit red. Is it red because it has some?
Peptone use.
Okay.
Because when the amino acids are being utilized, even when the amino acids are being utilized here, folks, this amino acid, the peptone by the deamination, and the peptones, when the peptone's carbon skeleton is used, there are two options. It can go aerobic respiration, or it can undergo anaerobic respiration. So even with the peptone, it can undergo aerobic respiration or the anaerobic respiration. If it undergoes anaerobic respiration, then you will see blackening in the butt area. If it undergoes the aerobic respiration using the peptones, you will see the red color.
Can you just write the reaction of the number 3? So it's the amino acid with the sulfur. The sulfur gets reduced to H2S, and that gives.
Yeah, the peptones, so peptones containing sulfur amino acid. Okay? So these sulfur amino acids, when they are used, it has to release amine group, it has to release the amine group, it has to release the sulfur atoms. These sulfur atoms which are given off, these are given off as H2S. And this H2S will immediately combine with the iron sulfate in the medium to form iron sulfide.
Is that why there's no gas in bubble presence? Because
It gets immediately converted to a hydrogen to iron sulfide. So Lisa, are you okay? Getting?
yes.
Yes.
In the atlas, it mentions that the black color comes from ferrous sulfate. How does that factor into all of this that we're talking about?
Because you have the iron sulfate in the medium, in the TSI medium, and H2S combines with the iron sulfate to form iron sulfide, and that's why you get the blackening. That's why you get the blackening.
But that's for number 3, not for number 1.
This one is for the number 3 and number 1,
Oh.
and this is also holds for good. But here the H2S is coming from the desulfuration. You can say just like the deamination, you're removing the sulfur atom. But this blackening, what you see here, is because of the anaerobic respiration. Okay? And that's why you see a red slant and the black. That means the bacteria is undergoing aerobic respiration here. But when it comes to the butt area, it is undergoing anaerobic respiration. And that's why when you're using the anaerobic respiration, the thiosulfate, Na2S2O3, that is reduced to H2S. This is the artificial electron acceptor in the medium.
So, to clarify, for the anaerobic respiration, when there's blackening in the butt, can we assume that there's a red slant?
Not necessarily, but then, it depends. Some bacteria you may see a red slant, some bacteria you may not see a red slant.
Okay.
Because if the bacteria is strictly anaerobic bacteria, then you may not see any color of the slant. The color of the slant will be just the original with no bacterial growth, but the butt area will be all black.
So for 3, it's anaerobic on the slant, and then aerobic
The
on
3 is aerobic on the slant, is aerobic respiration on the slant, but there is no bacterial growth.
I see.
The red is only because the ammonium hydroxide is simply coming down.
Gotcha.
So red means there's no bacteria.
red means no anaerobic bacteria. Folks, don't get too stressed out. Just simply relax, think about it, and then you will get it. You will be able to connect the dots that way. This is no rocket science. Believe me, it's not a rocket science. It's pretty straightforward. Think about it, don't get too stressed out. Take a deep breath, and then just think about it, you will get it. Yes.
So if we've got red slant, but yellow butt, we could conclude it's just glucose.
It is when you have a red slant and a yellow butt, it is because of respiration. It's because of respiration, but sometimes there's a balance between the acidic and the basic compound. When you see a yellow butt, okay, and the red slant, it indicates, because that's the thing what you have in one of the bacteria when you inoculated it, okay, is because it's undergoing aerobic respiration, but when it goes through the Krebs cycle, there are a lot of intermediate acids are being produced. And it is making those acids, and that's why the pH is turning to yellow color.
Oh, okay. So still all three sugars are being fermented.
It's still glucose utilization, and oftentimes if you see the red color of the slant and the yellowing of this one, that means they're utilizing the peptones. But the peptones, once they're utilized, once it goes into the Krebs cycle, the Krebs cycle intermediates are acidic intermediate, and that's what is giving me the yellow color. It was very strange. I saw it only once this time.
And how would you interpret these results in a biochemical explanation?
Oh, it's so easy. If I give you this one, that's the easiest one.
Okay.
This one, peptone use, anaerobic respiration. This one, fermentation of the lactose and the sucrose. Here it is just the peptone aerobic respiration.
That's it.
Oh, that's it. That's why I'm saying that you are all smart. You will get it. You will nail it down. I don't believe anybody will be falling behind. You are just too stressed out. Yes.
Can you go over the red slant, the yellow butt one more time?
I'll go. Okay, why don't I do one thing? Why don't I go with the respiration part, and I finish up this one, and I promise that you'll have a better idea. How about that? Okay?
Yeah.
And remind me, even if I won't forget it, but even if I forget it, remind me to get up on this one, and I'll make sure that you will get it.
Are you gonna put cell pictures on?
Pardon me?
Are you gonna put like cell pictures?
The similar type of pictures out there. Did I show you this one? There could be something like this. It could be something like this, or this, So I can put up this one. You never know this picture if this is there on the exam, like red color of the TSM medium, which is in the middle tube, is due to what?
No, that would be A and D. But that's not a question.
Huh?
Because it uses glucose no matter what, right? Glucose is always used.
Glucose is always used, but the red color is because of what? Glucose does not generate any basic. So it is high pH and the peptone use. Because when you use the peptone, you are not going to create a low pH, but you are going to create a high pH because of the deamination.
See, that's the tricky question.
So deamination.
Oh, that's the easy question.
No, sometimes it's tricky for us because we believe it's one.
Oh, no, you have to be thorough.
So deamination is connected to high pH?
this way, because deamination, you're removing the ammonia, and when the ammonia group is removed, it's a basic compound. Ammonia is highly basic. Oh boy, that was the chemistry part. Okay?
So it's not gonna be like cell system. It's
Huh?
gonna be more like yeast type instead of like bacteria cells.
The bacterial cell, I may show you a bacterial cell with a gram stain. And then I may ask you whether it is gram positive or gram negative, something like that.